Alright, ladies and gentlemen, this is going to be the next video in biology, specifically going over DNA review. Let's get started on this next video. So if we're going through and reviewing DNA is deoxyribonucleic acid. It's a self-replicating material which is present in nearly all living things and living organisms. And it's really going to be the carrier of genetic information and that genetic blueprint. So if we're going through and thinking about this, DNA is going to be the blueprint which is going to go through and create the proteins and the solid structure in our body. But it is the information carrying molecule that allows us to go through and create the organismal structures that we see in animals, plants, prokaryotes, fungi, all organisms that we see on Earth. Now, if we continue upon reviewing DNA is a nucleic acid, and it's going to consist of 3 parts. Each one of our nucleotides, which is the monomer that makes up our DNA, RNA, etc., has a phosphate, a pentose sugar, and a nitrogenous base. And this is what's going through and making up the nucleotides in that DNA. Now, we're also going to take those nucleotides, we have purines and pyrimidines, which we're not going to go through and review, and we're going to create that double helix when we go through and talk about DNA, and make it into that structure that we recognize and see as the familiar structure here. Now, in order to best understand this, we have to understand the hierarchy that we've also gone through and talked about before. Nucleotides are going to come together and they're going to make that double helix, or they're going to make a single RNA molecule. We'll talk about that later. But nucleotides are going to go through and create DNA. That DNA is then going through and condensed into chromatin. And chromatin is just winding that DNA around each of these histone molecules in order to go through and get it to compactly fit in the nucleus. So we can see that blue line is still the DNA, but we're wrapping it around these histones. Now, when we go through replication and mitosis, we're going to go through and compact these histones really closely together. We're going to wind that DNA really, really tight. And then what we get there is a chromosome, so that way it can go through and be separated via mitosis or meiosis, and we can get 2 separate cells. So we're going from nucleotides to DNA to chromatin, and then lastly to the most supercoiled chromosomes. Now, when going through and reviewing this, we have to understand Chargaff's rule. Now, Erwin Chargaff finds equal amounts of adenine and thymine and cytosine and guanine in organisms. And this is how we go through and come to the conclusion that we have DNA in the structure that it is. Now, yeast, beef spleen, and bacteria can all be gone through and ground up. We can blend all of them up, and we can go through and extract the DNA from it. If we go through and break down the DNA into individual nucleotides, what we're going to see is there's going to be an equal ratio of thymine and adenine. We're also going to see that equal ratio of cytosine and guanine, and this is what Erwin Chargaff went through and found. So Chargaff's rule is the rule that in DNA there is always equality in quantity between the bases A and T and between the bases G and C. This is important for us to understand when going through and doing a variety of other processes that we'll talk about in this unit. So to continue upon this, what we can see is G is always going to go through and match with C, A, and T, and we're always going to have equal ratios. And what we can see is each of these double helices are going to be held together via these hydrogen bonds. And these hydrogen bonds are what go through and allow us to understand how DNA forms. Now again, Chargaff's rule, it's equal amounts of A to T and G to C, so we can see that in the structure there in the diagram. And DNA is going to form in this double helix, moving in an anti-parallel fashion. Now, what do I mean by the word anti-parallel? Well, anti-parallel strands are strands of DNA double helices in opposite directions. And because of the opposite orientation of the sugar molecule, we see this anti-parallel feature. Now, this is important because Chargaff's rule again explains this process and explains how DNA is wound. Now, I want to make sure that we still emphasize the anti-parallel point here. What do I mean by anti-parallel? These are strands of DNA double helix in opposite directions. So let's look at this. We know that G and C are going to go through and bind together, and A and T are going to bind together. So if we go through and we look at the arrangement here, we have blue, green, orange, pink. Forget the A's and the T's and the G's and the C's right now. If we go through and rotate that diagram 180 degrees, flip it around, we can see that we have blue, green, orange, pink if we're starting from the top down to the bottom. If we were to go through and rotate this, we would see that they're the exact same strands, but the orientation is flipped 180 degrees. This is what I mean by anti-parallel. Now we're again back talking about these hydrogen bonds, and why are the hydrogen bonds important? Remember, a hydrogen bond is a weak bond due to polar interactions between molecules. But we can go through and replicate DNA by breaking these weak bonds and separating out these different sections, the two strands of the DNA. Weak bonding allows the DNA to uncoil for replication. It's really important that we have this hydrogen bond in between and not something stronger like an ionic bond in order to go through and get replication to occur. This is the process that we're going to go through and talk about shortly after this video. Now the hydrogen bonds, if we go into this and understand this just a little bit more, what we're going to see in replication is we're going to go through and separate the two anti-parallel strands of DNA via an enzyme. Since those hydrogen bonds are relatively weak, we can separate out those strands, and then all we have to do is we know that A and T go together and G and C go together. So now I can use these two strands as a template, create two anti-parallel strands with separate enzymes, and I know if I have that template, I can fill in the gaps there. This is how DNA replication occurs, and hydrogen bonds are the key principle within that. And there was a lot in this video, but I want to make sure that we understand the things that are necessary for review. Did you review nucleic acids, what the monomers are, what the polymers are, and the different nucleotides are? Did you go through and review the parts of the DNA? Do we understand how these parts of DNA go through and come together? Do we understand the hierarchy of DNA? And then lastly here, do we understand and are we introduced to some concepts like anti-parallel and hydrogen bonds, which are going to be key concepts for replication? This is the end of the video. We'll see you all in class tomorrow.